welcome to this video on the biosynthesis of phytohormones. Phytohormones are essential chemical messengers that control almost every aspect of plant life, including growth, development, and responses to the environment. In this video, we will explore how different phytohormones are synthesized inside plants, what their precursors are, and which enzymes and pathways are involved. By the end, you will have a very clear understanding of how plants produce hormones like auxins, cytokinins, gibberellins, abscisic acid, and ethylene. Let's begin by understanding what phytohormones are and why their biosynthesis is so important. Phytohormones, also known as plant hormones, are tiny signaling molecules that play major regulatory roles throughout the plant's life cycle. They control processes such as seed germination, root and shoot growth, flowering, fruiting, stress tolerance, and even aging. But, however, these hormones are not randomly formed. Their biosynthesis follows specific metabolic pathways, and most of them originate from common biomolecules like amino acids, fatty acids, lipids, and isoprenoid compounds. The pathway used determines the type of hormone produced, and the rate of biosynthesis helps the plant adjust its growth depending on internal needs and environmental conditions. With this foundation, we'll now look at how major phytohormone is synthesized, starting with auxins. The first phytohormone we will look at is auxin, specifically indole 3 acetic acid or IAA. The primary precursor for auxin biosynthesis is the amino acid tryptophan. Auxin is mainly synthesized through the indole 3 pyruvic acid pathway, also called the IPA pathway. In this pathway, tryptophan is converted into IPA and then IPA is converted into IAA. There are also some alternative minor pathways such as indole 3 acetamide or IAM pathway and the tryptamine or TAM pathway. But the IPA route is the dominant one in most plants. Now let's take a closer look at the key enzymes that carry out the IPA pathway. The first committed step in auxin biosynthesis is catalyzed by a group of enzymes known as TAA and TAR aminotransferases. TAA stands for tryptophan aminotransferase of Arabidopsis, while TAR refers to tryptophan aminotransferase related enzymes. These enzymes convert tryptophan into indole 3 pyruvic acid or IPA by transferring an amino group to alpha ketoglutarate, producing glutamate as a byproduct. This reaction marks the beginning of the IPA pathway and directly determines how much substrate is available to ultimately produce IAA. After IPA is formed, the next crucial enzyme family takes over, the YUCCA, flavine monooxygenases. Genes encode a family of flavine-containing monooxygenase enzymes or FMOs. These enzymes catalyze the key rate-limiting step in auxin biosynthesis, meaning that their activity has the biggest influence on how much auxin is produced in a plant. Yucca enzymes convert indole 3 pyruvic acid into indole 3 acetic acid through an oxidative decarboxylation reaction. This is the second step of the IPA pathway taking place after TAA and TAR enzymes generate IPA from tryptophan. Because they control this major bottleneck step, yucca enzymes play a very important role in shaping oxygen level in plants. Yucca enzymes are essential because they regulate local oxygen production inside plant tissues. Increasing yucca expression leads to an increase in oxygen levels, which can dramatically affect plant morphology. For example, overexpression of yucca genes has been shown to cause long hypocotyls, curled or epinastic leaves, and other auxin overproduction phenotypes. On the other hand, if yucca function is disrupted, serious developmental defects can appear in roots, shoots, and even during embryogenesis. The yucca family contains many genes, including YUC1 to YUC11 in Arabidopsis, and their tissue specific expression allows plants to finely control auxin gradients during growth. Next, we will move on to the biosynthesis of cytokinins. The next major class of phytohormones are cytokinins. Cytokinins originate from adenosine phosphates such as AMP, ADP, and ATP, and their major biosynthetic route is the isopentanylation pathway. In the first step, the enzyme isopentenyl transferase or IPT adds an isoprene unit to adenine nucleotides to form IP ribotides, which you can see at the left side of this figure. These IP forms can then be converted into trans-zeatin and cis-zeatin ribotides through additional enzymatic steps. For example, you can see here CYP735A enzyme hydroxylates IP to form trans-zeatin ribotides. The log enzyme then removes the ribose phosphate group to generate the active cytokinin bases IP, TZ, and CZ, shown at the bottom of the figure. Finally, cytokinin levels are regulated by CKX enzymes, which degrade these molecules by removing the side chain, ensuring that cytokinin activity does not exceed what the plant needs. Let us know more about the CK nucleosidases and CKX enzymes discussed in the earlier slide. CK nucleosidases, also called cytokinin nucleoside hydrolases, play a crucial role in cytokinin activation. They convert cytokinin nucleosides into free cytokinins, which are the biologically active forms. A good example is the conversion of trans-zeatin riboside to trans-zeatin, which we saw earlier. This reaction activates the hormone so the plant can use it. Many cytokinins move around the plant in their inactive nucleoside form, and nucleosidases enable rapid activation wherever cytokinin activity is needed in a specific tissue. Once cytokinins become active, they can also be broken down when no longer required, which brings us to CKX enzymes. CKX enzymes irreversibly degrade cytokinins by cleaving the isoprenoid side chain, producing adenine and aldehyde as inactive products. This is the main pathway for cytokinin breakdown in plants. CKX plays a major role in regulating cytokinin levels during development and stress. Overexpression of CKX leads to reduced cytokinin levels, resulting in smaller meristems and earlier senescence. In contrast, loss of CKX function causes cytokinin accumulation, producing enlarged meristems and delayed aging. In Arabidopsis, this enzyme family includes CKX1 to CKX7. After cytokinins, the next important phytohormone group we will discuss is gibberellins. Gibberellins or GAs are synthesized through the terpenoid or isoprenoid pathway beginning from the precursor GGPP, geranyl diphosphate. As shown at the top of the figure, inside the plastid, GGPP is first converted into N-copallyl diphosphate by CPS and then into N-choline by Ks. N-choline is transported to the endoplasmic reticulum where it undergoes stepwise oxidation by Ko and KAO enzymes to form Ga12, which serves as the primary precursor to gibberellins. From Ga12, the pathway continues in the cytosol. Ga12 is sequentially processed into intermediate GAs, including Ga15, Ga24, Ga9, and Ga20. Before Ga3-OX enzymes produce the major bioactive GAs such as Ga1, 3, 4, and 7. This figure here illustrates how GA biosynthesis spans multiple cellular compartments like plastid, ER, and cytosol, and how different oxidases control the formation of various GA forms. Now that we have covered auxins, cytokinins, and gibberellins, we'll move on to the next major phytohormone class in the upcoming slide. The next phytohormone is abscisic acid or ABA. ABA is not synthesized directly from isoprenoid precursors like many other hormones. Instead, it is produced from carotenoids, which are pigment molecules located in the chloroplast. As shown in the figure, the biosynthesis begins with the MEP pathway producing isopentenyl diphosphate, okay, which is converted stepwise into carotenoids such as zeaxanthin, anthraxanthin, and all trans violazanthin. Through the action of ZEP, these carotenoids are converted into 9-cis violazanthin and 9-cis neoxanthin. These are then cleaved by NCED enzymes, generating xanthozin, the key intermediate in ABA formation. Xanthozin is transported into the cytosol then, where SDR enzymes convert it into abscisic aldehyde, and AAO enzyme then converts abscisic aldehyde into ABA. This is a multi-step pathway spanning the chloroplast and the cytosol both. This allows ABA levels to rise rapidly under environmental stress conditions such as drought and salinity. But to understand this regulation more clearly, let's now take a closer look at the NCED enzyme family, which catalyzes the rate limiting step of ABA biosynthesis. So, what are NCED enzymes? NCED stands for 9-cis epoxy carotenoid dioxygenase. These enzymes catalyze the key rate limiting step in ABA biosynthesis, as said earlier. In other words, NCD determines how much ABA is ultimately produced because it performs the crucial oxidative cleavage that produces xanthozin from 9-cis carotenoids. The stronger the NCD activity, the more ABA a plant can synthesize, especially during water stress. Let's now explore the function and importance of NCDs in greater detail. Just to repeat once again, ABA is synthesized from carotenoids, you know that, and carotenoids are located in plastids, and NCD enzymes carry out the critical oxidative cleavage step that converts 9-cis violazanthin or 9-cis neoxanthin into xanthoxin. After this, xanthoxin travels into the cytosol where it is converted first into ABA aldehyde and then into ABA. Because NCD controls this key step, okay, it becomes the main regulator of ABA biosynthesis in plants. NCED activity rises sharply under drought, salinity, or other stress conditions, helping the plant increase ABA levels. Overexpressing NCED genes leads to elevated ABA and improves drought tolerance, while mutations in these genes reduces NCED activity and causes problems in seed dormancy as well as other stress responses. After ABA, the next major plant hormone is ethylene. Ethylene is a gaseous plant hormone synthesized from the amino acid methionine, and its production follows the Yang cycle. As shown in this figure, you can see it. Methionine is converted into S-adenosyl L-methionine or SAM by SAM synthetase. SAM is then transformed into ACC, 1-amino cyclopropane 1-carboxylic acid by the enzyme ACC synthase. ACC is finally converted into ethylene by ACC oxidase, releasing carbon dioxide, hydrogen cyanide, and water as byproducts. Meanwhile, the Yang cycle regenerates methionine through a series of intermediates such as MTA, MTR, and MT1 phosphate, allowing the plant to maintain a continuous supply of methionine for ongoing ethylene production. Because the cycle is tightly regulated, ethylene levels rise rapidly in response to developmental cues and environmental stresses such as fruit ripening, leaf senescence, and mechanical damage. With this, we have explored the biosynthesis of all the major phytohormones and regulatory enzymes that control them.